to us about lichens. I certainly do like lichens. I took only one course from Clifford Wetmore at the Itasca Biological Station, and I was talking to them about the fact that I still have my specimens. They should probably go somewhere. So anyway, that's about all I know about lichens, and I've not even read the book. James started his academic career, I would say, at least when he was age 13. He was helping out as a volunteer at the Academy of Natural Sciences in Philadelphia, where he was born. And for some reason, he was looking at these two lichens. And I'm reciting everything that I found on the web, which I hope is true. He was looking at these two lichens, and they were labeled differently, but they sure looked the same to him. And so he worked at it and figured out they were indeed the same, and one was mislabeled. My understanding is that he also had his first academic publication when he was in high school. And I was going to add up how many publications he has in his CV, which is 21 pages. You can go to the New York Botanical Garden, his webpage, and find his CV there. I counted back very quickly to 2012 when he got his PhD, and I was already at 100. And the first publication listed goes to 2000. So there's pages and pages more. So he's truly distinguished in his publication record. He also has a number of books. He has some new books coming out on lichens. We just heard at dinner about the Delmarva Peninsula, which would be Atlantic coastal area, which would apply to Cape Cod and other areas sort of north and south from there, so you can watch for that. And he's also done one on the Great Smoky Mountains also. So it's not just articles, it's also books. And I don't know how he does all that he does, because what I found in reading about him is he does scanning electron microscopy, and he's done it for years. He does, the regular taxonomic stuff that you do with chemicals and so forth. He does DNA, molecular work. He's collected and identified over 55,000 specimens of cryptograms only, that he says 90% are ID'd, they're ID'd, curated, and databased of the 55,000. He does not entirely ignore vascular plants. He has admitted to having collected 600, more than 600 vascular plants. And they were talking about vascular plants at dinner, so he says it's just a side hobby, vascular plants, or a side hobby. He's really focused on the eastern U.S., but he's also traveled in lots of other places and helped out in lots of other places in the world. So you can just talk to him later about his California and Colorado and other connections. The favorite thing that I found on the internet is the very last thing in his CV, which is the fact that there are two lichens named after him. And one is a sterile crustose lichen that's endemic to the Galapagos Islands. And it's the genus Lapraria, which is also the genus of his dissertation. And then the other one named after him is a fungal parasite on a crustal lichen, and I didn't even know there was such a thing. But he's also done some interesting lichen naming. Again, you can go to his New York Botanical Garden webpage, and there's a link. I haven't opened it yet, but it says Dolly Parton something, but he and his student have named a very, I don't know what would be the right word, anyway, a lichen that's appropriate. It's a lichen, right? Yeah. Okay. A lichen that would be appropriate to name after Dolly Parton. and that's a true story. And it's on his webpage. So James, he's at the New York Botanical Garden now. He's the assistant curator of lichens. He also, where he did all his graduate work, what's it called, City University of New York.
University of New York.
He's still connected with that, and there's a cool course taught out in Colorado. I saw the lichen biome. So anyway, he's done a lot of interesting things. And we're thinking if you, all right?
If you don't learn my password.
No. Thank you again for being here, and I'm sorry you had to wait almost 2 years to give your talk.
Thank you. Thanks. Everyone can hear me, right? I don't usually have a problem with that, but I always like to double check before I actually start to talk. So thanks for the introduction, and I would really like to say, as Nancy had, that I really appreciate sort of being brought back after my potential to having given a talk like this 2 years ago. This is a talk that I've really wanted to give. This is not the talk I would have given 2 years ago, I assure you that. But it actually has a lot of other information in it as well. But it's really a topic that I really thought was really appropriate for an NEBC meeting and a New York Flora meeting. And so it kind of saddened me that what happened did. So it's really a pleasure to be able to come here and do that. And I'm really excited to be able to do it. It's really something I have firmly enjoyed putting together this talk, and I hope you all enjoy it as well. I hope it's as much fun for you as it was for me. Let's make sure that's on. That is now on. So the other thing is that I'll share with you that my most exciting thing that has happened today or on the trip so far is that I spent the day annotating specimens in the Farlow Herbarium, and I was looking at specimens that Edward Tuckerman and his friends collected in Cambridge in the early 1800s. And back in those days, they would reuse paper that was from whatever they had gotten at around the time. And one of the packets that I opened was made out of a broadside for John Quincy Adams for governor.
Wow.
Which, I mean, it's just, I'm from Philadelphia. I worked in a historical herbarium for many years, and it's the kind of thing that you can just only find there, and it's just such a gem. I mean, you can imagine this, like, somebody handing it to him on the street, or it was, like, plastered to his door, and he ripped it off and folded it and made a packet out of it and put a specimen in. And now we have it for, posterity today. So it's kind of amazing. I really, really love it. So anyway, I am here to talk to you about the lichens of New England and really northeastern North America. I don't like to just limit it to New England per se. We all have different definitions of what New England is and what northeastern North America is. But what I'm really going to focus on is sort of talking about the rich history of lichens and lichenology that we have here in northeastern North America, and how we can use those resources to really move forward with doing really interesting things with these dynamic organisms. So before I go into the details of all of that, I always think it's helpful to sort of give a little bit of an overview about what lichens are, why they're important, mainly because it's amazing and I just like to be able to tell this to people, but also I think it just helps everyone sort of appreciate and understand them better. So lichens are cryptogams, and cryptogams are, by definition, plants in the historical sense, so that's plants that include fungi, that reproduce through spores rather than with flowers or seeds. And so the word cryptogam, that means hidden sex or hidden reproduction. And so these are things which hundreds of years ago, when scientists that didn't have DNA sequencing technologies and smartphones, when they first went out into the world and were classifying nature around them, these were the botanical organisms where the reproductive structures were either too small to see or too difficult to discern. So they didn't really understand what was going on with their reproduction, and they grouped them together. The result is, of course, that now we know that cryptogams, in that historical sense, includes lots of different organisms that aren't necessarily closely related evolutionarily. So it includes things like ferns and fern allies. We all know and love those, I'm sure. It includes mosses and liverworts. Some of us love those a little bit more, or a little bit less, depends on your proclivities. It includes things like algae, so your red algae, we used to include blue-green algae, now those are cyanobacteria, those are not even closely related. And of course it includes fungi and lichens, and you'll notice that there's a lot more pictures of those, which probably tells you where my true love and proclivity lies. Maybe you shouldn't tell my spouse in New York. Anyway, so I'm a lichenologist and I focus on lichens, fungi in general. So when we're talking about cryptogams, we're really, not talking about the sequoias and the pandas of the world. So that means that you actually have to sort of pay attention to the trees in the forest. You have to see the trees for the forest. What's growing on the bark, what's growing on rock, what's growing on soil underneath your foot. It means diving through a hand lens and looking up close at things. And really there's this amazing dynamic world that exists that is created by cryptogams, or populated by cryptogams. Now, even though these things aren't necessarily closely related to each other, they often form tight-knit communities and grow together. And so you can see here, this is just a sort of piece of a trunk of a tree in the mountains of western North Carolina and the Great Smoky Mountains. You can see here there's a fern. This is an epiphytic cryptogamic community. Here's a moss, there's a liverwort, and there's a lichen. There's, of course, more than one lichen, more than one moss, more than one liverwort in that picture. But what I like to sort of appreciate and take away from this is that these 4 different cryptogamic groups that aren't necessarily closely related to each other grow together in these really tight-knit communities, on other things, of course, as well. So lichens are what I study, and that's what the rest of this talk will be focused on. If you want to learn more about bryophytes or algae, I suggest you find someone else to come and talk. What are lichens? Lichens are a lifestyle. I've trademarked that phrase. They're a lifestyle that's evolved multiple different times in the fungi, wherein fungi form symbioses with algae and cyanobacteria for the purposes of obtaining nutrition. So this is, and these 2 things that look very different from each other get together and form a body, a thallus, that looks completely different from if they had grown independently in nature, which to me is one of the most amazing things about them. So this is the thallus, or the body, of Anaptychia palmulata. It's a foliose lichen that was once common and widespread throughout temperate eastern North America, and it's something that's actually suffered declines over the last century, and I'll be talking a fair bit about conservation and overall in this talk. And so that this is a species that now tends to be associated with mature forests and more intact natural habitats throughout its range. It's also endemic to eastern North America. If you were to cut through that lichen, make a section, and look at it under a microscope, what you'd see is that it's dominated by fungal structures. So there's a fungal reproductive structure, and then there's these lovely layers of fungal hyphae up here and elsewhere. And then there's also a layer of algae inside, and those algae are photosynthesizing, and they're providing sugars and things for the fungus. And so this is a fungus and an alga. They're in a symbiosis. They're working together to do this. They're lichens. Lichens are a lifestyle, arguably the best one. So if there's one thing that probably most of you have heard or learned about lichens at some point in your lives, if you're not a lichenologist, then you have probably encountered them in basic biology in either college or high school, and you've heard that they come in 3 main forms. So these are forms that are just sort of a way that we conveniently discuss their sort of how they grow or what their structures are, but they are not necessarily all closely related to each other. So the first form are these fruticose lichens. Those are the big bushy ones, things like British soldiers, which a lot of people are familiar with, or things like old man's beard. Those are fruticose lichens. They're 3-dimensional, they're shrubby, they grow away from the substrate. They don't have distinct upper and lower surfaces. And as you can see here, this is another example of 3 different fruticose lichens, and you can see that they attach to the substrate more or less in one place, and they hang down or they grow up. They're very 3-dimensional and bushy. Your second kind are your foliose lichens. Those are the leaf-like ones. They have a distinct upper surface and a distinct lower surface that are 2 different colors. You can see that really nicely here in this Parmetrema reticulatum. The upper surface here is blue-gray. The bottom surface is brown. They're like leaves. And what I like to sort of, I like to put these slides up to show people that, don't try to pay attention to the Latin names. They're written small so that most people can't read them. I certainly can't with my bad eyesight. But I like to put this up so that people appreciate that there's this amazing diversity of colors and forms that are sort of in the lichen world. And that really, I think, really gets to the point that they're not all closely related to each other. It's lots of different fungi that have entered into this lifestyle. So these foliose lichens, they have this foliose morphology where you can see that there's these lobes down here with a distinct upper and lower surface. Again, if there's one thing you should take away from today, it's that you'll be able to identify a foliose, fruticose, and crustose lichen. So crustose lichens are your last kind, and those are the ones that are firmly attached to the substrate. They have an upper surface, but no distinct lower surface. They're the things that look like paint on trees or rocks. They're in intimate contact with the substrates. They're also extremely diverse and extremely understudied compared to the macrolichens, and that's a point that I'll really hone in on today. So when we talk about lichens, we're talking about symbioses, but the important thing to realize is that we're actually talking about fungi. So these are species of fungi. The reason for that is that when you're talking about lichen species, the only member of the symbiosis that is consistent everywhere the lichen grows is the fungus, or at least the primary fungus that forms the lichen. So this is Ramolina stenospora. You can wave to it if you would like. This would be its name tag at a event or a mixer. And so everywhere this little shaggy lichen grows throughout the southeastern United States, it's formed by the fungus Ramalina stenospora. Now there's a chance that everywhere that lichen grows, it associates with the same alga to form the lichen. So that and maybe everywhere it grows, it's Trabuxia jamesii. But there's also a chance that it might associate with one or another alga, depending on local conditions that are more favorable to another alga. So maybe Trabuxia jamesii is what this lichen fungus associates with in the mountains of, in the coasts of North Carolina, and maybe Trabuxia glabra is what it would associate with in southern Florida, and maybe Trabuxia lichenum is what it would associate with in sort of the bayous of southern Louisiana. The point is that is why they are named based on the fungus. It's because the fungus is the one consistent member of the association. Sometimes it's a one-to-one relationship with the alga, but not always. And algae, of course, have their own names.
Do they look different if they have different algae? No?
Depends. Not usually, but as with everything in biology, sometimes. There's only one exception, one sort of case of that, where they look really different. We can talk about that later. It's actually pretty remarkable. So if there's, again, something else that's really important to take away from lichens, it's that they form these symbioses, and these symbioses are extremely important and critical to terrestrial ecosystems. All of them, all of the terrestrial ecosystems on this planet. So they're extremely important as ecosystem pioneers, so they are some of the first things to grow on soil, on bark, on rocks, and they break down the rocks to form soil or are involved in the formation of soil. They're also important in nutrient cycling, so things like the nitrogen cycle. They contribute substantially to nitrogen cycling in forests, and that work was actually done in eastern North America and in the Pacific Northwest. They're also extremely important, and I think underappreciated in this way, for climate regulation. So one of the things that makes lichens particularly interesting and unique is that they have this ability to dry out and become effectively metabolically inactive for long periods of time. And then as soon as any water comes near them in any form, be it as fog or rain or pee from a moose, they will suck up that water multiple times their dry body weight. And then instead of drying out rapidly, they release it slowly back into the environment. And now that might not seem like anything if you're talking about a lichen that's the size of your hand, but at the scale of an ecosystem where you have metric tons of lichens growing on trees and rocks and everything in profusion, you can imagine how that has the effect of regulating the environment so that there aren't extreme swings in temperature and humidity. And that's really important to the small-bodied fleshy invertebrates that form the bottom of the food chain. It's important for the germination of seedlings, all sorts of things like that. Of course, lichens are used by lots of large animals. I don't take very many pictures of large animals because I scare them away when I'm in the field with my hammer and chisel. So you'll have to suffice with a picture of the boreal forest where the reindeer would have been. But reindeer survive on them for 80% of their diet in the winter. Flying squirrels and squirrels use them. Many birds use them for nesting materials. Lots of animals use them for their diets or for their lifestyles. Small, mid-sized, and small animals use them as well. One of my favorite examples, because it involves the dust lichens that I studied for my PhD, are these lacewings. Lacewing larvae put little sort of coats of dust lichens on their back, and that it is useful for camouflage, right? So the things don't eat them. Also, they sort of stay hydrated, and so they help keep that little fleshy invertebrate that's sort of young and immature cool and happy and moist. And then when they metamorphose into an adult, they make their little capsule out of the lichen that's on their back, and then they pop out as adults. It's actually pretty amazing. I have a colleague who did take one and go through its whole life cycle on his desk, so he has pictures of all the different stages. That's what it looked like when it popped out. And then finally, so you go from the giant animal to the smaller animal to really the smallest scale. And that's within the lichens themselves. There's this whole universe that exists absolutely nowhere else in nature. So there's like lineages of bacteria, fungi, and algae that exist absolutely nowhere else. There are nematode worms. There are other invertebrates like tardigrades, water bears. If you have not appreciated the wonderful world of water bears, I strongly suggest that you Google them not during this talk. But they're absolutely amazing. So really, what I hope you take away from this introduction is that lichens are truly hubs of trophic interactions, the same way that coral reefs are. They are the coral reefs of the land. I would argue that there is, almost nothing that happens in a terrestrial ecosystem that doesn't in some way probably involve a lichen. They are so incredibly dynamic with the things that go on in them, around them, between them. It's just truly amazing. I mean, once you appreciate it, you just sort of can't stop thinking about it. It's actually kind of crazy. And that something so important could be sort of so underappreciated by the public at large is just shocking to me. We should all be snorkeling in coral reefs of lichens with our hand lenses. So now I'd like to sort of turn to what the more sort of focus of this talk is, and I want to start with a little bit of history because I think this is certainly germane to northeastern North America. So the field of lichenology actually has its start at the same time as early botany in North America. And so that's really with, I would consider the grandfather of lichenology to be in the United States and Canada, to be Henry Muhlenberg. Many of you may know Henry Muhlenberg. He's a fairly well-known botanist from the Philadelphia area. He lived in Lancaster County, Pennsylvania. And what Muhlenberg did is he was actually an amazing scientist, amazing botanist. And in the late 1700s and the early 1800s, he went all around his home in Lancaster County and traveled around in southeastern Pennsylvania and basically conducted what we would today consider to be biodiversity inventories. He was extremely effective at collecting all the organisms around him. He's particularly well known for vascular plant collecting, but he did a really good job with lichens too. And unlike many of his predecessors, he didn't focus on sort of big, showy, conspicuous macrolichens. He really made an effort to collect everything, including the teeny tiny little crusts. It's actually amazing that somebody in the late 1700s was doing that. It was actually pretty unusual at the time. So he generated this amazing collection of things, and then he actually tried to figure out what they were and guessed that many of them were new, but he then sent them en masse to the father of lichenology, the whole field, Erik Akarius in Uppsala in Sweden, and he classified them and named them there. And so the result is that many of the most common and widespread species, including also rare species as well, in eastern North America were named by Akarius in the early 1800s based on specimens that were collected by Henry Muhlenberg near his home in Lancaster County, Pennsylvania. It's a good story because it means that we know where they came from and we can go back there today to see what's still growing there. So if Henry Muhlenberg is the grandfather of lichenology, then without a doubt the father of American lichenology is Edward Tuckerman. And you should all know that name. If you don't, Mikhail School might come after you later. She'll have a thing to say about that. I certainly would too. He's the one who had the broadside for John Quincy Adams. So Edward Tuckerman spent most of his life working in Massachusetts in Amherst, and he started his career collecting lichens in the White Mountains of New Hampshire. That's some of the work he's particularly well known for, but really he ended up devoting his entire life and career largely to lichens. He studied other things in the botanical world, but he's particularly known for lichens. So he worked in the White Mountains. Tuckerman Ravine is named after him. So is Tuckerman Brewery, the Tuckerman Brewing Company. You should write them and tell them that they need to commemorate him and his lichen legacy in some way. I tried and was unsuccessful. And of course, he's well known for having named British soldiers, which is one of the sort of iconic fruticose lichens that many people know and love, and has these lovely red fruiting bodies. And so Tuckerman produced 4 major book-length works, which is pretty astonishing for that time. He published the first summary of the lichens of New England in 1848. He published a book on the lichens of California and an appendix that included basically lichens from other places that I may one day want to visit in 1866. And then he compiled the first lichens of North America, the first synopsis, in 1886 and in 1888. 1888 is after he died. It was actually posthumously finished. It's a 2-volume work. And then in 1872, he also published the first sort of generic summary of all the lichens of North America. That's a huge amount of work for someone to do, in addition to just sort of generally classifying and dealing with describing the world and the biodiversity that was around him. So I really think that this should highlight to you that going back to the American Revolution and before, there is a basis, a body of literature, a body of knowledge about the lichens of northeastern North America and of New England. And so that should really give you an idea that over the centuries that have elapsed since that time, a lot of data have accumulated. And so the lichens, the data for lichens in northeastern North America is really not so shabby. And I think a lot of people might actually be somewhat surprised to see a map like this. So this is a map of all of the geo-referenced and database specimens of lichens from the northeastern North America in the Consortium of North American Lichen Herbaria, which is sort of a giant consortium where we have all of the lichen herbaria data pooled for North American herbaria. And you can see that's 45,000 plus collections that are mapped there. That's a pretty good number, right? And you can see if you zoom in on Massachusetts and the surrounding states, that again, there's a lot of dots there. So I put this up there because I think a lot of people might be surprised to learn that there are this many records of lichens. So every one of those dots represents at least one specimen having been collected somewhere. Apparently, people like to go to Cape Cod. But what's important to sort of, all of those dots hide something, and that's the fact that just because there's a lot of dots don't necessarily mean that there's a lot of collections from where those dots are. So if you just look at the state of New York, there's 30,000 plus collections of lichens that have been made in the state of New York and databased, and of those only 8,000 are georeferenced, so they have a latitude and a longitude associated with them. But you can clearly see if you map those that they are concentrated in different portions of the state. So here's a heat map of those collections, and you can see that the red areas, which are where there was 265 collections made, per grid cell. I forget what the exact grid cell size was in this map, but the main point is that basically people have collected lichens primarily in the Catskills, the Adirondacks, Long Island, and southern New York. I don't think many people would be surprised to learn that. But it really highlights that there's vast areas of this state that have been virtually uncollected for lichens. And I'm sure that if you added those additional collections that haven't been georeferenced to that, it would not change this pattern very much. Maybe you would get another sort of hotspot near Buffalo or something like that. But lichens tend to be collected around cities or in places where people like to vacation. Now the other thing to take away from this is that there's also, so we have a lot of data, that's great, and there's a great baseline for future work. But the amount of data that we have is nothing compared to vascular plants. So what you see here are the different states in northeastern North America. Their names are too long to do anything other than abbreviate. And on the top there, you see the numbers of specimens of lichens that are in these databases, this big database of North American lichen herbaria. It's not a perfect system, so maybe few have been missed, but I think these numbers are pretty representative of reality. And then you can also see the numbers of plant specimens. What I'd like to highlight is the number of times more plant specimens there are than lichens per state. You'll see that Connecticut, particularly, has 23 times more plant specimens collected than lichens. I don't know, I was actually kind of shocked to learn this, that there's 205,000 collections of plants collected in the state of Connecticut, and yet only 8,000 lichen specimens. That's pretty amazing. So again, these numbers aren't perfect. It doesn't represent every single specimen ever collected, but they're a pretty good representation of reality, I would say. It's a little bit more striking when you put it on a sort of bubble chart like this, where the mustard color are the plants and the green are the lichens, because again, they're prettier, so they deserve the nicer color. And you can see that there's quite a disparity between those things. So we have data for lichens, but it's certainly nothing on par like we have for vascular plants. And I think that's a problem. Now another thing that I'd like to highlight is that we have this great body of data that we could do something with, and we should be doing something with, and in some ways we are. But one of the real problems is that it requires a lot of work to actually use it. So I think that if you study plants, if you study vascular plants, if you went into a herbarium, like if we left this meeting right now, we walked into the Harvard University herbaria, and we said, I want to look at specimens of, let's just say, lady slipper orchid, Cypripedium macaulay. I think that the chances are that if you went and you opened a folder that had Cypripedium macaulay written on the outside of it, pretty much every specimen in that folder probably would be Cypripedium macaulay. If not, it might be another species of Cypripedium, so it might be something closely related. I bet the same would even be true of many things that you would think of as being difficult groups of vascular plants, like Carex. You know, sure, they might not all be perfectly identified, but I bet there's a pretty darn good chance that if you opened a folder that was had Carrick species, whatever you want it to be, booty eye on the outside of it, most of the things that are in that folder would probably be more or less right. Well, that's not the case for lichens. And the misidentification rate or the incorrect identification rate by modern standards in most herbaria can exceed 50 or 60%. And the reason for that is, trust me, I've done the numbers, at least in my own herbarium. The reason for that is that whereas vascular plant taxonomic concepts have remained more or less stable over very long periods of time, that can't be said for lichens. And so I will give you two examples of that. One of them is this lichen, Parmelia conspersa. This is a name that was applied to pretty much every green foliose lichen that grew on rocks historically. So here you can see this is you might not be able to read this, but this is a collection from Lake George made in the 1880s. And then here's another specimen. Oh, gee, one collected by Edward Tuckerman in New Hampshire. And you can see this is a name that was applied quite widely over a long period of time. So if you were to just follow if you went to the checklist of North American lichens and you said, well, I have a specimen that was collected in 1889 and it's called Parmelia conspersa, and I'm going to put it in its right place, you would update it to Xanthoparmelia conspersa because that's the change that's been made. That's what it's currently recognized as. That's great. But you would have probably a 50% chance that it's actually Xanthoparmelia plittii, which has the same chemistry, the entirely same range, and just the only difference is the color of its underside. You'd have a maybe little less probable chance that it's Xanthoparmelia verriculo-umbrina, which again has the completely same geographic distribution and just differs in its reproductive mode and chemistry. And then you'd have an even lesser chance that it's this other species, which again is entirely sympatric and simply has a different chemistry and different reproductive mode. The point is that you would actually have to physically look at the specimens, do chemical spot tests on them, which are quite easy to do, but you have to do them nonetheless, look at them under a microscope, and then decide which of these four things they were. And that's assuming that it was right to start out with. In my experience, many specimens collected in the 1800s can be, especially the early 1800s, can be radically misidentified. Not through any fault of the people that were working back then, it's just that concepts have changed so much that something was called one name back then, and now it could be 10 species in 4 or 5 different genera. This is a concept that I think is very foreign to people that work in vascular plants. And it's why using these data are really, really difficult. It requires a lot of work to go and look at them. Another example is Acrolechia parella. This is a name that was really widely applied well into the 1950s for pretty much every species of Acrolechia that grew on rock or bark in North America. In our region here in northeastern North America, you would now have a choice of 4 different species. So if you and incidentally, Acrolechia parella doesn't occur in North America, so that just makes it even harder, more difficult. So if you were to update that name, what would you update it to? You would have to look at specimens. But the worst part is that not doing this, not taking the time to do this, is a problem because these species have very different distributions and ecologies. So Acrolechia mexicana, that's a northern species. Despite the name, I know it's really strange. It's actually particularly common in the Great Lakes region. I didn't do it, not my fault. It's actually quite rare, it's northern. Acrolechia pseudopalescens, that's something that's more temperate and it's much more common. Same thing with Acrolechia tropophora, that's more temperate and common. And then you have Acrolechia yasudi, that's actually pretty rare in northeastern North America. It's made more Appalachian and coastal. So again, if you didn't change these names and actually look at them, that would be hiding all of this information, which is a huge problem. Another thing that I think is really endemic to lichens, and it has received very little study, and anyone here, actually Elizabeth Lightberg, you were asking me about this earlier, things that you all should be looking for. Here you go. So in lichens, we have this really weird case of species that appear to have been described and then completely forgotten until modern times. And it's unusual. I don't think that that's the case in vascular plants, certainly not on this scale. So my colleague Aaron Tripp at the University of Colorado and we went through the entire checklist of North American lichens, which has 5,400 species on it, and we scored a bunch of character traits for a study we were doing. Would you believe that 500 of the species on that list, we could find no information on their geographic distribution whatsoever? Those were mostly names that had been used once historically. Many of them were described from North America and have never been used since. Are those real species? Are they species that have been gone extinct? Are they species that are just masquerading under some other name? We actually don't know. It takes a lot of work to work that out, but those species are potentially the ones that are the highest conservation concern, or are the most interesting or unique for some weird way. So one example of this, I'm going to give you several because I just thought it would be really enlightening to see some of these. I think it's quite surprising. They illustrate different aspects of this problem. This is Catalaria glauco nigricans, which was described by Edward Tuckerman. Here is a picture of its thallus. That's from the type specimen in the Farlow herbarium, which I looked at. This is a section of its apothecium. Those are its asci and its ascospores. It was originally described by Tuckerman based on a specimen collected by Henry Willey in New Bedford in Bristol County, Massachusetts. Now, it subsequently was reported from all over North America, but nowhere else really in temperate eastern North America, which is quite strange, I think. There's even a description published of it in the Sonoran Flora in 2007. You'll notice where it says that it's just characterized by its granulose thallus. It doesn't have a granulose thallus. That's the actual type specimen. So I looked at the type specimen. One person has ever looked at it before, nearest I can tell. It was annotated in the 1980s. And so that lends the question, first of all, why has no one ever collected this species again in the area where it was named? Is it rare? Is it extinct? We don't know. But then also, what are all the other collections that have been called it elsewhere in North America? They're probably not the same thing, and they're probably a different species. But this is a case of something that's just been completely forgotten, more or less. And to me, it really highlights there's something in this region that is described from here. It's very distinctive. It's quite a distinct species. I've never seen it. I've never collected it. Collected 56,000 specimens, named a lot of them. Never saw this one. So where is it? Is it extinct? People ask me the question of whether there have been extinctions in lichens all the time. And my answer is we don't really know. Well, here's a case where maybe we do. If you went out and did targeted searches for this and made targeted efforts to look for it in herbaria and in the field and you didn't turn it up, maybe this is an example. And this is a good way to look for examples of things that may have gone extinct, narrow local endemics that went extinct before they were really detected in any meaningful way other than someone by pure chance collecting them over 100 years ago and then them getting described. Another example of this, which again, I think is quite interesting, was Lichenology willii. This is a species, this is the type specimen collected by Henry Willey, and actually out in Amherst, not on the coast. And it's a pretty distinctive species. It was described by Tuckerman in 1882 based on one collection from Amherst, Massachusetts, and two collections made in southern New Jersey. I borrowed them, looked at them. It's got a very distinctive morphology. It has a very distinctive chemistry, unlike anything else. I've never seen the species. I spent a lot of time in southern New Jersey. I'm from Philadelphia, and I never saw this. So is this species extinct? Is it very rare? We haven't detected it. What happened to it? Where did it go? It's missing. It's forgotten. But they're not all just things like that might actually represent distinct species that have either gone extinct or are extremely rare and have potentially really high conservation value. The reason that I highlight this is that this is an example of a species that was misunderstood. So this is a species, Lichenia virginiensis, described by William Nylander, based on a collection from West Virginia, from a town that actually doesn't exist anymore. It's kind of interesting, small mining town. And so this is a picture of the actual type specimen. For some reason, so it was described in the late 1890s, and subsequently it was reported from all these different places in temperate eastern North America. Only one author, or two authors, ever really used that name. One of them was John W. Thompson. I'm not quite sure why he particularly liked this name, but he put it on a lot of specimens, particularly in Indiana, and in Wisconsin, and in Michigan. Well, it turns out that when you look at the actual specimen, it really belongs to Bryobylindea ailesii, and that's the distribution of Bryobylindea ailesii in North America. The type specimen falls well within the geographic distribution of that species. It's clearly identifiable as it. But then that lends the question, what was all the other stuff that was called Lichenia virginiensis over the past century? So it's just a case where this one was misunderstood. They're not all potentially these sort of forgotten species that may have gone extinct or might be quite rare and of a high conservation concern. So we have these historical data, and there's really a lot of work that needs to be done to do something with them. But that doesn't mean that we can just rest on our laurels and sit in museums all day and look at specimens. Someone actually has to get out and do the field work, the boots on the ground work, to really generate modern baseline data to compare with those historical baselines. And the really fortunate thing is that we have people that have been doing that. And there are people in this audience, two of whom are illustrated here. It just so happens both of their names are listed. They have been quite active in this regard. There's a whole group of people that have been active in New England, in the Boston area, in New York. Here is just a map of specimens that were collected by Elizabeth Lay, who is here. And this is 5,300 of them where the identifications have been verified. The specimens have been digitized, georeferenced, and they're now available online and in the virtual herbarium in the New York Botanical Garden. But it really illustrates to you that there is actually work being done, but more work is needed. Now another point that I would like to make is that macrolichens are by far better studied and collected. And I think anybody that knows something about lichens would tell you that, but it's really, really crazy. So macrolichens are the really big, showy ones that everybody knows and loves, like your British soldiers and your old man's beard. I'm partial to them as well. I fully appreciate that. Your foliose lichens, your fruticose lichens. So if you look at the total number of specimens collected for all of northeastern North America, 66% of them are macrolichens. So out of 153,000 specimens that were collected in that whole area, or sorry, 15,000. Oh, wait, that's right. Anyway, the number might be off there. 66% of them are macrolichens. Oh yeah, the number's off. It should be 150-some thousand. It's about the same for all of the states within northeastern North America. So you can see that macrolichens are much, much better represented in collections. Does that mean that crustose lichens are less diverse? Does anyone want to guess? Well, so first of all, clearly there's been a collection bias strongly, strongly towards these conspicuous foliose and fruticose lichens. I don't think anybody would be surprised to hear that. These are the things that you could easily walk up to a tree and just pick off with your hands. And they're just more showy and conspicuous, so a lot of people would have collected them all over time in the past. So microlichens, in fact, are, of course, understudied, but nonetheless, they're as diverse. So here, this is the summary of the lichens of New Jersey, where I compiled all of the historical reports from New Jersey. So I went through every single published paper on the lichens of New Jersey, and then also all the data that we've accumulated at the New York Botanical Garden, which has the largest collection of New Jersey lichens. I did a lot of work there when I was in undergrad. And so we ended up with a checklist of 473 accepted species, 248 species are published reports, prior published reports that were excluded or questionable. So that actually highlights that, again, a large number of the previous published reports actually someone has to go back and look at the specimen to figure out what species they refer to. Chances are it's one of these 473. It's a long story, but just take my word for it. You can feel free to debate me about that later. Of that 66% is pretty much a 50-50 split between macros and micros. So that's scary. Then you can go to Indiana and do the exact same thing. There's actually a lot less published data for Indiana, thankfully. And so I did the exact same thing, went through all the published reports, evaluated them for whether or not they were sort of reliable and likely correct, questionable, or totally not likely to occur in Indiana based on published distributional data, or having actually gone to the specimen myself and said, oh, it's misidentified. There you go. So again, you can see it's about 25%. That's actually a similar number. It's kind of interesting. So of those 221 species that occur apparently in the state of Indiana, again, it's basically a 50-50 split. So two different areas of eastern North America of different sizes and with different numbers of diversity, and it's more or less a 50-50 split both times between macrolichens and microlichens. It should be extremely this should be extremely distressing given the extreme disparity in herbarium collections. and the fact that pretty much all ecological studies and many studies of lichens in North America have been focused on macrolichens under the assumption that their distribution patterns, their biology, their ecology follows that of microlichens. So basically, many fields of lichenology, at least in this country, have developed nearly absent the perspective of something that represents half of the biota. That's crazy. So this is why what I say, what really is needed, and this could be, I mean, we have this amazing body of data, and something could be done with it in a way that was really amazing and really transformative for the field and for biodiversity sciences in northeastern North America and really the United States and Canada in general. You know, I think that what we need is a large-scale systematic inventory, and this is a sort of idea that I've come up with. It's not new or anything, but it's something that I've implemented in other parts of the United States, in the coastal plain and in southern Appalachians. What we need is a large-scale inventory that builds on the work that people have done in the recent decades to sort of build and establish a modern baseline, but take it further and expand it and really sort of really make it bigger. We need to have standardized sites that are uniform in size and habitat, distributed throughout northeastern North America. You need to carry out lichen inventories of those sites where you actually collect vouchers of the specimen of all the species there, bring them back to herbaria, deposit them, identify them, you know, do all the things you would standardly do that as part of that. We need standardized habitat quality assessments of those same sites. We also need to have information about the forest structure that is pretty easy to generate even in a relatively short time frame. We also need to do things like quantify the rockiness and the logginess of the forest, again, to quantify more things about the habitats themselves and for potential correlations with the lichen diversity and the lichens that occur there. Same thing with woody plant diversity, and even if we want to tackle it, herbaceous plant diversity. That's just a lot more time intensive and hard to do on a large scale. And then finally, I think that what we need to do is to incorporate into that environmental sampling so that we have a potential to begin to understand the symbiont pools that are present in the environment. So we know that lichens are obligate symbioses between fungi and algae and cyanobacteria and other things. But what we don't, and we understand where those lichens occur, more or less, on the landscape. What we don't understand is where are their spores? Where are those symbionts? Are they just everywhere ubiquitously in the environment, or are they much more narrowly distributed? Why do the lichens only grow where they grow? Why do the rare species only occur where they do? You know, are the spores everywhere and they're just not able to grow there because of some abiotic condition, like it's too dry or it's too hot or something? Or is it because the alga is not there? The fungus can make its way there and the spores land, but the algae aren't there. There's so many questions that could be answered with that. And of course you can look at then changing things with historical baselines and everything. I mean, there's a lot that could be done. So what I'd like to do is sort of run through 2 examples of where we have done some really interesting things with historical data, going and doing more work in modern times, and then actually done something with conservation as a result of the findings that we had. So the first sort of example or case study that I'm going to tell you about involves an old man's beard, Usnea. So this is a species of old man's beard. You can see why it's called that. It's because it looks like a beard hanging from the tree. Maybe that gives you a little bit of a better impression as to why it has that common name. I was a lot younger in that picture. So this involves this species that's called Usnea angulata. It has this common name, fisherman's dream. You'll hear more about that in a second. It's a species that it grows meters in length and it hangs down from the canopies of trees. If any of you are familiar with Usnea longissima from the Pacific Northwest or from the boreal forests or other parts of the world, beautiful species. It sort of drapes down from these trees. It's just absolutely beautiful. Usnea angulata does the same thing. Different species, same kind of idea of how it grows. It's distinctive and extremely distinctive because its branches are ridged and have these angles. So it actually looks like the backbone of a fish. That's why it has that common name. If there was one lichen or one old man's beard that a fisherman would dream about, it's this one. It's the catch of the day. So this is a species that's so beautiful that even botanists who aren't specialists in lichens collected it. So it was actually first collected by Henry Muhlenberg in Lancaster County near his home in I think 1803. Sent to Eric Acarius and named as new to science. So it was named based on this collection from Pennsylvania. This is a collection from, this is not that one, this is a collection from the Palisades in New Jersey, and I just put that there to give you a sense that this thing really is not just like a little tiny lichen. This is a standard herbarium sheet, and you can see that the lichen has been sort of bent multiple times. This thing's big and showy. And so you cannot be surprised to learn that pretty much any time botanists saw it, they were sort of so awed by it that they put it in a press, probably, and brought it back. And so there's actually a fair number of historical records for it. This is the distribution of the species in North America prior to 1935. After 1935, there were no collections made. So we have a pretty firm idea of where the species occurs. This is a distribution pattern that you pretty commonly see in lichens. We actually went back and borrowed almost all of the specimens that were the basis of these occurrences and examined them to make sure that they were correctly identified. The species is very distinctive, so it's not surprising to learn that, in fact, the vast majority of them were correctly identified. That was really nice to see. But again, it's just to highlight that a lot of work went into producing a map that has some dots on it to tell you what the distribution of this species was prior to 1935. So then we went out and we went to many of these places to see whether or not this species was found there. And of course, we were looking also for modern occurrences in herbaria. And so what we found is that this was the occurrences prior to 1970, really prior to 1935. So there was this gap from 1935 to 1970 where not a single new occurrence was found, or no specimen was collected, or nothing was known. This is where it occurs now. And we can say with a fairly high degree of probability that this is where the populations are now, or the subpopulations are now. The thing is quite distinctive. People know what it looks like. Basically, almost all of the populations occur in the southern Appalachian Mountains, primarily around Great Smoky Mountains National Park. There's a small subpopulation in the Ozarks and the Ouachitas, and there's one around Leech Lake. All of them are in old-growth forests. All of the lichens grow on conifers. Why might the species not occur throughout the rest of its historical range? Because all of those forests probably got cut down, and that species is probably highly dispersal limited, and so it was not able to recolonize those areas once the forests grew back. We knew nothing about the ecology of this thing from the herbarium specimens, and we only found out about how it grows, the fact that it has this really weird growth form like Usnea longissima, which actually doesn't firmly attach to its substrate. So most Usneas, old man's beards, attach by a point to the substrate they grow on. These species don't. So they just drape from the tree and they fragment and fall into lower branches and then keep growing. And then as the tree grows up, they grow up, and more fall down. And so they tend to be concentrated on one or two trees in a small area, and you find smaller numbers of individuals sort of moving out from that area. Anyway, this is its currently known distribution. So it has declined since at least 1935, we know, and that's about in a span of 3 generations approximated for lichens. We have rough approximations for generation times that are used, and that's a decline in its total extent of occurrence of 65%, and its estimated area of occupancy by almost a third, or almost a third. That's quite a lot. And it's actually probably much more drastic than that. Those are really overestimates. If you know anything about how those values are calculated, I will promise you those are overestimates. That's assuming 2 kilometers of suitable habitat around a point, and there are not 2 kilometers of suitable habitat around most of the places where the species occurs. So I told you it occurs on mature conifers. Guess what the conifer is that it primarily occurs on?
Hemlock.
Yep, of course. So if there's anything you know about hemlocks in the southern Appalachians, it's that most of them are dead. And one of the reasons why we find this species particularly easily these days is that as the hemlocks have died in places like Great Smoky Mountains National Park, where many of you might have gone to see the fabulous old-growth hemlocks that were there, now the lichens that are growing in their canopies come crashing down with the branches and the canopies, and that's why we find the lichen, because it falls down. So here's the green in the trees is the Usnea angulata, again here. So this is in Great Smoky Mountains National Park near Cataloochee, a place many people have gone as tourists and probably loved. It's a beautiful place, but unfortunately many of the trees are dead now. This is in the Nantahala National Forest in the Chattooga River Wilderness. Again, there's still individuals of the lichen there, but many of the host trees have died. This highlights a clear problem. And it's something that could be done about with immediacy, right? So a threat was clearly identified, and we propose an action. rather than just let the lichens rot on the ground and contribute to the nitrogen cycle, what we did was we went around and we started to pick up the lichens wherever we find them and brought them back to Highlands Biological Station. And it turns out Jessica Allen, who was my former PhD student and is now a professor at Eastern Washington University, she found that these are quite easy to grow and propagate. You just cut them and put them on wooden dowels, drape them down from trees, and they grow just fine. And so that's what we've done. We started doing this with our lichen class at Highlands Biological Station 2 years ago, and she's been monitoring them and adding more ever since. And so what we've done is tried to create at Highlands Biological Station a nursery, more or less, of these Usnea angulatas that are derived from populations all around the southern Appalachian Mountains with the idea of trying to protect whatever genetic diversity was there. And with the idea that if we continue to propagate them, there's a chance that we'll be able to reintroduce them into the environment when maybe the adelgid that's attacking the hemlocks goes away, or maybe if the adelgid, doesn't really kill all the hemlocks this far north, there's a chance we could do translocations and bring the species back up this far north. So something could be done, but the point is that now they're at least alive and something can be done with them moving forward. Here's another picture of my former research assistant putting them up in the tree. You need someone particularly tall to do that. Jesse and I are both short, so it's not very helpful in that regard. Then you have to get them down again, which I guess is why you have to continue to hire the research assistant. So then the last little vignette that I'm going to present to you involves this lovely lichen, Cladonia submitus. It's a reindeer lichen, and it has the common name beach broccoli because it grows on beaches and looks kind of like broccoli, I guess. And so this is a species that I think many of you might actually be familiar with or have seen. You might not have known you've seen it. But it grows in coastal dunes on Cape Cod, Long Island, and in southern New Jersey, and also on Delmarva. And so here you can see there's a map of all of the known occurrences from prior to 1990. And the red are all the places it was known to occur after 1990. So those are, we noticed when we first did an assessment of this species that it appeared that there had been a decline. But this is a really common, very conspicuous species. And we thought, okay, so this is what appears to have happened. But we'll put it forward for IUCN red listing and potentially for Endangered Species ESA, Endangered Species Act protection. So we made a proposal and everything, documenting its decline. But this proposal was tabled because we thought that we didn't have adequate population level data for modern times. So we know kind of where it occurs, but we wanted to really have a good estimate of the number of individuals and a really firm idea of where it occurred, given that this species occurs in the mid-Atlantic, it's endemic to here, and it occurs in highly visible and important areas to people where they recreate with, there's a lot of things that go along with that. If you have a species that's very abundant and common in some places on Long Island and Cape Cod and in southern New Jersey, you really want to have a firm, robust sort of backing before you propose something to protect it, really. You want to know that it really is threatened and that it is in decline. And so my new PhD student decided to take this on as a project, and he actually went and did a complete population assessment of this with funding support from a number of different botanical clubs and organizations. And so this is him in the field. By the end of the project, he really liked Cladonia. And so what he did is he went and he actually looked at went to every site where the species was historically known and where it was known from modern times and assessed whether or not it was still there, and then estimated the population size. What he found, which is kind of interesting that we didn't really know before, is that this species is a species of sort of stabilized coastal dunes in coastal areas. And it needs natural dunes. So anywhere where the dunes have been screwed up for one reason or another, this species is no longer there. We'll get to that in a second. This is in Island Beach State Park in New Jersey. This hosts the largest remaining population of the species, which is basically one continuous colony on dunes for 2 miles long. It's pretty amazing. Nonetheless, what his study showed is that, so this is everywhere the species is known to have ever occurred, and this is where it occurs now. And so that is a nearly 50% loss in the number of locations since 1990 or more, which is crazy, right? So if you run this through the IUCN red listing criteria, this is a clear decline, and it's a clear decline that ranks it as endangered. So there's actually like a really identifiable charismatic macrolichen that grows in our local area that is endangered. And we have the data to support this. Something can be done about it. What are the threats? Well, I think the threats would be pretty plainly obvious to anyone that's ever been anywhere along the Atlantic coast of eastern North America, and that's really sort of destruction of coastal dunes and large-scale development, largely in the past because there's a lot of regulations now that would prevent certain things from happening. But this is I show this because this is a site where the species occurred at in 1936. It was collected actually right about here. This is Pikes Beach on Long Island. This is what it looks like now. This is from Google Earth. It's amazing the things you can do in Google Earth. This is what that looked like in 1994. 1994. That species, there is definitely suitable habitat in here for that species. That means that the species was probably extirpated from this site within my lifetime. It's not like this is something that happened long, long ago and we can sort of fault the ignorance of history. This is something that happened in modern times, and it's something that is ongoing. So it's something that could clearly be done about that. The other thing that I think is really important is that this shows that, that this work didn't just die with this. It doesn't just end with having shown that this species is in decline and we need to do something about it from a conservation perspective. There's also some really interesting things about cladonias. And so this work allowed Jordan to go around and do population level sampling for detailed population genetic studies of all the cladonias where he worked. So all these places where he went and sampled, he did these transects and sampled all the cladonias growing in these colonies where these cladonias grow. If you look at a patch of a cladonia, you'll see that it's not just one species growing there. There's actually, in this case, in North Carolina, there's 3. Cladonia arbuscula, Cladonia rangiferina, and Cladonia, and a new species, potentially. We actually don't know what that one is. It's kind of crazy. And so, because of this work, he is going to be able to actually infer the population genetic structure of this rare and endangered species and also characterize the community of cladonias that grow around it. And that's really important, and put that in the context of the broader cladonia world in eastern North America. So what I'd like to sort of end on is to just summarize the things. There's a lot of them I know that I've just talked about, and I'm happy that you all made it through here with me. So I would just, first of all, data do exist for lichens. They are not as abundant as for vascular plants, but that doesn't mean that we don't have a lot of data that are not extremely useful, or at least potentially useful, if we review them and do things with them in the right way. We also need to collect more data, and we really need to do that in a large and concerted and systematic fashion. The biodiversity science is something that has happened in sort of fits and starts over time, and it's often thought of as an individual pursuit. And one of the things that we really need to do moving forward as a community is to recognize that these sorts of things have to be systematic, large-scale community efforts with sort of maintained consistency and standards so that everything can be compared. Do you know how hard it is to compare the numbers of species of different things that occur at a place? Have you ever tried? It's really difficult. So to find out the number of lichens that grow at a state park, you might make a list of the state park lichens, but then there'll be a list of the birds for that county, but not that state park. And then the vascular plants, well, maybe they'll be for half of the state park. And then maybe for mammals, it'll be for just the rock outcrops in that state park. It's not completely overlapping, so you can't really tell something about all the different things. You can't compare them. It's really difficult. So I hope I've showed with at least the 2 examples that, and the various species that are potentially lost or that have been misunderstood, that there is possible to understand these things. And that, it doesn't just end with understanding them, that more science happens after that. This is really the beginning of addressing really interesting and compelling hypotheses about biology and ecology and things like that. There's just so much that can be done once the house is in order. And then finally, I really like to highlight that conservation of these things is A, needed, and B, possible. It's not that we don't know they're threatened and we don't know what to do about that. Something could be done if one was so motivated. And so, thank you. Maybe you need to buy that book now. Maybe I convinced you to up your offer.
Any
Is there time
questions?
for questions?
Oh, yeah.
I might have gone on for way too long. I'm sorry.
I have a,
Is that fair?
no, but I have several, but I'll just ask one. So you're in going back and reevaluating with the chemical studies.
Hmm?
As botanists, I don't know. Do you write down your chemical analyses, annotate with those?
If you do it right, you do.
Okay.
Yeah.
And yeah, so that was my. Does everybody who wants to annotate it have to test it? But you. Okay.
Depends who you trust.
Okay.
Yeah, more or less. I mean, no. Once it's been done, if you trust it and it looks right, often the spot tests remain. Well, some of them remain, so you can look at them and say, oh, it looks more or less like it would have been. Yeah. I think if there was a reason to do it again, you would, but we try not to do that. We understand that to actually look at something involves sectioning it and doing a spot test or looking at the spores. That is destructive sampling of specimens. So, that's care is taken, and it's just one of those things that you understand as someone who works on these organisms that, one, you have to collect specimens that will be usable in the future for people to look at, and two, that you have to be very careful and sort of thoughtful with museum specimens that exist, and that's just something that you have to learn as a student over time and get better at, Does that answer your question?
Absolutely. Yeah.
Anyone else?
One of the commonest types of lichen in Massachusetts seems to be umbilicarius. I wonder if you have any comments on that. Are there just they all look like this relatively one or two species to me, and could there be any rare ones here?
Oh, I'm sure there's a whole raft of things that are rare. Well, there's two that might not even be around anymore. I mean, that's the thing, the place to start is that it's really interesting to me that there's Henry Willey and Edward Tuckerman and others in Massachusetts were really good at collecting things a long time ago. And a lot has changed since then. And so if there's a place where you would go to start to see what's rare and what's not, it's go back and look at that stuff and see what's not here anymore or what hasn't been recollected. And we have lots of people, some of whom are here in the audience, who would serve as an excellent resource for that purpose. So yeah, I mean, the commonest things are going to be pollution sensitive and disturbance tolerant lichens. Those are like a cohort of 20 species or so that occur on like every street tree or tombstone or something like that. It's great they're coming back though. They were gone from most places in the 1970s. I wasn't there to see that, but they were gone at that time, but the air quality has improved, and so a lot of things have come back. But I think that so much so that people are now afraid they're hurting their trees. But the thing to take away from that is that they've come back, but what has come back are really disturbance tolerant species, because basically everything is one forest edge now. And so it's the things that can tolerate those forest edge habitats. The shifts that you see in lichen communities from the 1830s or from the late 1700s to present day are remarkable. I mean, there's species, the things that we have that are on like every rock and tree, some of them there's no collections of prior to 1930 or something like that. Whereas they're the only ones you see now. And in contrast, there's this huge amount of stuff that they used to get everywhere, that they used to find routinely. And you can read floras that say from 1890 that say common, or from 1930 that say common, or even some of them will say rare, but they're still there. They were documenting them, and those things are have never been found here in the last 100 years. So, yeah.
When were the first lichens? When did they first evolve?
We don't really know the answer to that question, but presumably a really long time ago. Because logically they would be, well, I mean, that's actually not a silly question or answer. Like, my answer might seem a little silly, but we actually don't know the answer because there's not a really great fossil record for them. So, like, incontrovertible fossils of lichens only go back so far, and yet there's a high probability that they existed well before that. And then, it's just philosophically, if you think about life colonizing land, something resembling a lichen would be a really logical form for terrestrial life to take early on in the colonization of land. I mean, you need food, you need a home, protection from the UV radiation. Let's get together and form something that looks maybe something vaguely like a lichen.
Each species would have had to evolve previously to the lichen, right?
No, not necessarily. What do you mean? They could just meet and form it potentially. The proto-lichen.
I mean, you had to have the alga,
Oh yeah.
the lichen,
And the fungus.
and
Yeah.
the fungus.
But they were around. Those go back a long time. I mean, there's marine fungi, there's marine algae, there's freshwater algae, there's freshwater fungi. They were around in that time, and at some point, some of them transitioned onto the land, and met up and never left.
But you have no numbers at all.
No, I'm not numbers that I'm comfortable giving, let's put it that way. There are numbers out there, but I don't like them. So I'm a skeptic at heart.
What about the new thinking on ascomycetes in fungus, or in lichens? Yeasts?
Have you, so this is a great story. So have you read the paper on that?
No.
So many of you have probably heard this thing that was all over the media about a year or two ago about this third symbiont that was found in lichens. Well, I think it's a really great, well, first of all, it highlights that there's this amazing body of work, and I never really get into this because it's sort of like a whole nother level, and if you're just talking about lichens to start out with, getting into the things going on in lichens is a whole nother world. But there's this whole body of work that goes back decades now where people have really done amazing work documenting these just super diverse communities of bacteria and fungi and algae and other stuff that lives in the lichens and around them. And there's been a lot of really elegant work done documenting that shows how amazing the communities that these things are. It's not just an alga and a fungus, it's more complex than that. But that paper actually didn't show that there was a third symbiont involved at all. So if you read the paper, that's not in there. Would you be surprised to learn that the media made something up and said something that was completely not representative of what was actually in the publication? I love it as an example, and I love because everyone asks, anytime I give a talk, I get asked this question, regardless of the setting. It's actually amazing because that media story penetrated so deeply into the public that it was forwarded to me so many times. But it's a really great teachable moment because, I have colleagues that are PhDs and you have formed an opinion about this, and did you read the paper? This is why you have to always go back and read the primary literature. You can't just believe what we read on the internet about me. You have to actually go back and read what the original source says, because that study is a really elegant study of the microbiome of lichens. It has this amazing fish staining that was done in it. It shows some really cool things, but it doesn't show that there's a third symbiont that contributes super importantly high important functions that was totally unknown, but it doesn't do that at all. It builds nicely on a body of work that's existed in a really collegial and scholarly community for decades, which is a better story, I think, but maybe not as sensational.
Are there any examples of introduced lichens?
Another great question, which again, the group of people here is particularly relevant to. So most lichens know we don't have examples of known invasive species, and so that's partly why lichens are like the ideal way to talk about the biodiversity crisis to members of the public, because when you try to explain to them what's going on in the world around them, and you talk about plants or animals, when native plants or animals go extinct or are extirpated or decline in numbers, invasive species come in. And so people see plants, they see animals, and they go, oh, everything's fine. And then it's a discussion that becomes very detailed and too detailed, and people's eyes glaze over and where they don't believe you. Lichens are very different. Look at a tree outside and you will see that there are not the lichens that were there 130 years ago, 180 years ago. It's very different. You go to an old growth forest and you go to a midtown Manhattan, and the difference is clear. There are no lichens, or very few in one place, and there are tons in the other. So that's a really important thing to recognize. Does that help? What was the rest of your question again? Sorry, I just got completely lost in my train of thought.
It was if there are any known
That's right.
mutations of introduced.
So they don't do that for the most part. That's why we lose them. But there are small numbers of them. There's a couple species that we know appear to have been brought in from afar and become established in like a city park or 2 city parks, but haven't really traveled outside of them in North America. Outside of North America, that's a different story, and it's thought that there might be 1 or 2 examples that are more, I wouldn't want to use the word invasive because it's nothing like Japanese stiltgrass or something like that. But we do have some that are not, a couple that are non-native, but they're not like expanding their ranges really. There's only 1 that we think might be doing that, and that hasn't been well documented, so I'm not going to present it as like hard science here. And it's pretty, so I wouldn't mind. I wouldn't mind having it like covering all the trees. That's fine. Let it be here. If we have too many lichens, then that's like a problem for another day. Anybody? Yes.
Without knowing much about it, it seems like I've read in places or abstracts that at least lichens that grow in heathlands and coastal areas, that they actually inhibit seed germination.
Not all of them. Some of them.
Not all of them, but
some
there are
facilitate.
some, and would that be like some of the cladonias that are in mud dunes?
I actually don't know that they inhibit, if someone else can correct me on this incidentally, there may be someone that knows more about this than I, but I don't know that they inhibit the growth of things. I mean, they actually haven't been demonstrated specifically to change the soil underneath, at least in Eastern North America. There is literature on that and studies on that in Europe that might be different, but I'm sure they do something, but I'm sure some of them have an inhibitive effect because they produce all these chemicals, and then some of them do other things. There are some plants that need the lichens present to grow, or at least the seedlings need them there to sort of, I guess, provide the right microclimate for them to survive. But the examples of that there's few of them that have been studied in detail. So I don't think a lot of people are going around looking to see whether or not the lichens are helping the plants grow. They're all just, killing our trees. They're not doing that, by the way, at all. Anyone?
Another dumb question. So
There are no dumb
they're named after the fungus.
Mm-hmm.
So that fungus, you wouldn't reuse that name. The fungus only grows in that relationship. It wouldn't.
Yep.
Okay.
Dolly Parton's lichen is only applied to that one fungus.
Okay.
Yep. And it forever will be.
All right. I guess if there's more questions,
I'll be here to chat.
we can take them next door in refreshments. But first,
You got a couple more minutes to throw something in. I have the highest one in my pocket, so you won't know what it is.
Is anybody interested in bidding on the book?
Anybody else interested in bidding? Otherwise, I'll just announce it right now.